Students, let us continue. You have already seen meta rotation. Next slide I am continuing from meta rotation, so there is no need any confusion. So we are seeing disaccharides on hydrolysis they yield 2 monosaccharide unit. Common example is sucrose, which is a table sugar which is obtained from sugar cane. Okay, you have seen that sugar cane this one common. So beetroot, which is sweet, carrot, honey, very sweet, so they are all contain sucrose. So sucrose on hydrolysis gives glucose and fructose. Sucrose on hydrolysis gives glucose and fructose. So what it contains, here one this composition is not clear. Glucose and fructose, down what is written is alpha D glucose and beta D fructose, so no confusion. Next to glucose plus fructose, what is written is alpha D glucose, which is unavailable. There is alpha D glucose and beta D fructose. I repeat, alpha D glucose and beta D fructose. So glycosidic bond is alpha 1 alpha 2, okay, alpha 1 alpha 2 glycosidic linkage, alpha 1 alpha 2 glycosidic linkage in sucrose. So alpha 1 alpha 2 glycosidic linkage, okay, alpha 1 beta 2 glycosidic linkage. So alpha 1 beta 2 glycosidic linkage in sucrose. So I repeat this. In this slide it is not so clear. So sucrose composition is glucose and fructose, which contains alpha D glucose and beta D fructose. What is the glycosidic bond present here is alpha 1 beta 2 glycosidic linkage, alpha 1 beta 2 glycosidic linkage, which can also be called as beta 1 2 glycosidic linkage. So you better write in the notes alpha 1 beta 2 glycosidic linkage in sucrose. Okay, so in maltose, which is present in germinating seed and malt, so it is also intermediate of digestion of starch and glycogen. Both are polysaccharide. Composition is glucose plus glucose. Again you see here what is the composition here of lactose is maltose. Composition is alpha D glucose. Both are glucose, no? So alpha D glucose plus alpha D glucose. So something is not unable to see there, that is alpha D glucose. Both side alpha D glucose, both are same in maltose. Alpha D glucose plus alpha D glucose, okay, alpha D glucose and alpha D glucose. So alpha 1,4 glycosidic linkage. So in maltose, alpha 1,4 glycosidic linkage. In this slide last sentence, glycosidic linkage is alpha 1,4 glycosidic linkage. So I want to tell you one thing. Glycosidic linkage again we will see later. So it is a bond between two sugar molecules, two in polysaccharide, for example, two sugar molecules linked by glycosidic linkage. Okay, so OH there is a linkage, and it is a covalent type of bond, glycosidic bond. So there is a removal of water molecule, so there is a bond formed which is called as glycosidic linkage bond. So why it is there? It held together two glucose molecules by covalent bond in polysaccharides. Okay, so bond present in carbohydrate is glycosidic bond. Detail we will see next coming slide. Short note is there for glycosidic link. So now you can remember that maltose contains two glucose molecules. The glycosidic bond is alpha 1,4 glycosidic bond. Lactose is milk sugar which contains galactose and glucose. Okay, lactose contains galactose and glucose. See there, which is unavailable is beta D galactose and beta D glucose. See this slide, something unavailable is beta D glucose and beta D galactose. Both are beta. Is it clear? Beta D glucose and beta D galactose. Okay. So glycosidic bond in lactose is beta 1,4 glycosidic bond. Beta is unavailable there. So beta 1,4 glycosidic bond. So beta D galactose and beta D glucose. So glycosidic bond in lactose is beta 1,4 glycosidic bond. So also disaccharides are classified as reducing and non-reducing sugar. You know what is reduction and oxidation reaction. So these sugars, maltose, lactose, isomaltose, they are reducing sugar. They act as a reducing agent. They for example, they Kipric to they convert Kipras. Okay, so they can share completely transfer the electrons. Okay, so that they get oxidized. They are reducing agent. They transfer the electron and they get oxidized. Okay, Kipric to Kipras oxide. For example, in Benedict test, in practical demo you will see Kipric to Kipras. So they are reducing agent. They share the electron, complete transfer of electron, they get oxidized. So they reducing agent, maltose, lactose, isomaltose. But sucrose is a disaccharide which is non-reducing. This reducing property because of the presence of free aldehyde or ketone group in monosaccharides. These sugars, because of the presence of free aldehyde and ketone group, they are reducing agent. But sucrose and tahallose is non-reducing. In sucrose there is no free aldehyde or ketone group. But sucrose on hydrolysis yield what? Glucose and fructose. So that again, if you break the sucrose into glucose and fructose in solution, again it is reducing agent. Otherwise you have to remember that sucrose is non-reducing. So these are the hydrolysis of disaccharide. Maltose gives glucose and glucose to isomaltose. With the maltase is enzyme, isomaltose gives glucose and glucose. Isomaltase is the enzyme. Lactose, glucose and galactose. Lactase is the enzyme. Sucrose, glucose and fructose. With mineral acid also we can hydrolysis. So, okay, this is the simple one what we see. So next is the polysaccharide. What are polysaccharide? They are carbohydrate which yield more than 10 unit of monosaccharide, either same or different. Like starch gives only glucose, but heteroglycans gives different, same or different. Okay, so let us see starch. You may get short note on starch. What is starch? It is plant storage homosaccharide. What is glycogen? It is animal storage homopolysaccharide. Okay, so made up of glucose molecule. Starch is made up of glucose molecule. It consists of two components, water soluble amylose and water insoluble amylopectin. Okay, so what are the two components of starch? Water soluble amylose and water insoluble amylopectin. So amylose is unbranched. Remember, amylopectin is branched. Okay, amylose is branched. Amylose is unbranched and amylopectin is branched. They contain about 400 to 1000 glucose unit held together by alpha 1,4 glycosidic linkage. Okay, helical structure it is. As I told, amylopectin is branched. The straight chain portion has alpha 1,4 glycosidic linkage. See, there is something unavailable, that is alpha. Okay, alpha 1,4 glycosidic linkage. See, in straight chain, what is the linkage? Alpha 1,4 linkage. Wherever branching point is there, alpha 1,6 glycosidic linkage. See, that unavailable is alpha. I repeat again, don't get confused in the slide. Alpha, both side alpha 1,4 and alpha 1,6 glycosidic linkage. Most starches contain 80 to 90% of amylopectin. Amylose is only 10 to 20%. That means they are branched. Okay, this is the small diagram which shows about your straight chain and branched structure of starch. So again, glycogen, as I told, it's an animal storage. Glucose, excess glucose, after a food intake, excess glucose is used for energy in the cell. Excess glucose is stored in the liver as glycogen. So it is the source of energy. Animal storage. Glycogen is branched. Again, straight chain portion, it resembles amylopectin because it is branched. So glycogen is glycogen resembles amylopectin of starch. So it is branched. But what is the difference between glycogen and starch? Remember, glycogen also branched, but glycogen is highly branched. It is a animal storage homopolysaccharide. It is a highly branched. So it is amylopectin. Amylose not there. The straight chain portion here, branched structure. Straight chain again branched. 1,4 alpha 1,4 glycosidic linkage, where at branches it is alpha 1,6 glycosidic linkage. It is highly branched and then amylopectin of starch. Okay. Starch is less branched, but glycogen is highly branched. It is mostly amylopectin. Okay. So that is the just a diagram to show alpha, wherever alpha 1,6 linkage, where it is alpha 1,6 at branching point. Straight chain, what is the linkage in? Is alpha 1,4 linkage. Next is the inulin. Inulin is a homopolysaccharide made up of fructose molecules. It is not much importance. It is exogenous compound which is used in a kidney function test, clearance test. Cellulose is plant fiber. Okay. Cellulose is a plant fiber. So cellulose is plant fiber made up of alpha. It is made up of beta D glucose unit linked by beta 1,4 glycosidic linkage. Okay, beta D glucose, where it is unavailable is beta D glucose unit. Cellulose is composed of beta D glucose unit. What is unavailable in this slide is beta. Okay, beta D glucose, that is molecule. So cellulose is most common dietary fiber present in the food. Okay, that is in the function of carbohydrate you have seen. Next, I just mentioned PPT2, but we started early this PPT2. So homopolysaccharide example, starch, glycogen, inulin. Heteropolysaccharide, they are polysaccharide, but on hydrolysis they give more than one type of monosaccharide unit. So mucopolysaccharides are best example. They are also called as heteroglycans, mucopolysaccharide or heteroglycan. See, these heteroglycans are glycosaminoglycan made up of repeating units of sugar derivatives, amino sugar and uronic acid. Both are sugar derivatives. Okay. Most of the GAGs means glycosaminoglycan contain sulfate group and some of them contain acyl group. Hyaluronic acid, chondroitin sulfate, heparin, dermatan sulfate, keratan sulfate, heparan sulfate. Okay. Hyaluronic acid is unbranched linear chain of alternating beta glucuronic acid and an acetyl beta glucosamine. See, remember, no need to write the name, but you have to write the tissue distribution and function in case short note comes. If you remember, you write the the structure in word, that is made up of unbranched linear chains of alternating beta glucuronic acid and acetyl beta glucosamine. So this is mainly present in hyaluronic acid in synovial joint. Remember, it is present in mainly in synovial joint. It acts as a lubricant. See, this fluid, synovial fluid deficient means there is a deficient of hyaluronic acid. So joint problems comes. That is why it acts as a lubricant. As in the machine, you have seen the lubricant is required. Okay. So chondroitin sulfate made up of alternating unit of alpha glucuronic acid and sulfated acetyl beta galactosamine. So cartilage, bone, skin, tendon, cornea, even blood vessels, it is present. It helps to maintain structure and shape of cells. Heparin is a natural anticoagulant. That is the important thing you have to remember. In heparin, it is a natural anticoagulant. Tissue distribution, blood mainly, okay, lung, liver, kidney, spleen. So heparin, second function, it also helps the release the enzyme lipoprotein lipase. This lipase enzyme, lipoprotein lipase, helps in clearing turbidity of lipemic plasma. Lipemic means if more triglyceride is there, in the blood serum, it appears turbid, lipemic. Okay, that is lipemic plasma we call. If more triglyceride fat concentration is there, this turn whitish, that is lipemic. So what is the function of heparin? It is mainly present in the blood as anticoagulant. This you have to remember, maybe a one word question. Okay, so also it helps in the release of the enzyme lipoprotein lipase, which helps in clearing turbidity of lipemic plasma. Next is dermatan sulfate. It consists of repeating unit of iduronic acid. This is important in dermatan sulfate. Even in heparin also there is a beta iduronic acid is there. In dermatan sulfate, repeating units of iduronic acid and N-acetyl galactosamine here. Okay, galactosamine. Blood vessel mainly dermatan sulfate, heart, valves, skin. Have a structural role in sclera, responsible for shape of eyeball. Okay. So keratin sulfate made up of beta galactose and N-acetyl beta glucosamine. Uronic acid is absent here, unlike dermatan and heparin. Unlike this here, uronic acid is absent. Mainly cartilage, cornea, connective tissue, intervertebral disc it is present. So main function of keratin sulfate is it keeps the cornea transparent. What is the main function of heparin? It keeps, heparin is a natural anticoagulant. Okay. The last one is heparin sulfate, skin, fibroblast, aortic valve, lung, muscle and liver is the main tissue where this heparin sulfate is distributed. Heparin sulfate binds to a variety of protein ligands. Ligands are small chemical molecules, okay, and regulate the wide range of biological activity. So heparin sulfate bind to variety of ligand, okay, and regulate variety of biological activities. For example, development processes like blood coagulation. Blood coagulation, clotting is required, okay, prevent bleeding. Angiogenesis. What is the meaning of angiogenesis? Synthesis of cell wall. This one. Blood vessel, synthesis of blood vessel, angiogenesis. So heparin sulfate plays a very important role in angiogenesis with the help of this protein ligand. So these are the some of the things about mucopolysaccharide. They are heteropolysaccharide. What are those? Heparin sulfate, keratin sulfate, dermatan sulfate, heparin, chondroitin sulfate, hyaluronic acid. What is the function of hyaluronic acid? It is present in the synovial joint, act as a lubricant. Okay. So others also there you can see. Homopolysaccharide example, glycogen animal storage, starch is glycogen animal storage, starch is plant storage. So starch contains two amylose and amylopectin. Amylopectin is branched. What is the difference between starch and glycogen? Glycogen resembles amylopectin of starch, but it is highly branched, okay, compared to starch. Okay, these are the some of the aspects. Okay, thank you.